Okay, what do we have going on over here? We got a bunch going on. We're going to be working with MALDI-TOF MS. Look it up. It's a long word. The MS, I think, stands for mass spectrometer. Okay, so this MALDI-TOF MS, we're going to be talking about what's going on in here first. So all these little dots right here are a colony, and the colony is added along with the solution, or you could say the solution was added to the colony. So colony plus solution equals matrix. So this whole thing right here is called a matrix. So first, a portion of the colony along with the solution called a matrix is spotted onto a sample plate. So this is my little sample plate that I put it on. So I went dot, 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 and I put it onto a sample plate. The sample plate, so here's my sample plate right here, is then set onto an apparatus. And I just drew a big square because I don't actually know what the apparatus looks like. So once it's set onto that apparatus, that exposes the sample plate to a laser beam. Bam, bam, bam. That laser beam is hitting that apparatus that's on that sample plate with that matrix, okay? And that laser beam is a treatment. And what it does is it converts, it both converts molecules to a gaseous state and it ionizes them. So not only is it a gas, it's an ionized gas, okay? So here's our gases. Right here, some of them are big and some of them are small. And you can see I wrote that they're ionized, and here I wrote little I's on there to know that they're ionized, okay? So what happens next? Well, this procedure with this laser beam, it's called a soft ionization. Oh, it's so soft, ionization. And why do they call it that? Because it leaves the large molecules relatively intact, okay? So it leaves those large molecules relatively intact rather than breaking them into small pieces. Then the ions then travel through a flight tube. So these ions travel through a flight tube. So here's my flight tube right here. It's traveling through that flight tube. And because small ions travel, because small ions travel faster than the larger ones, so the small ions here, that's why the small ions are first, small ions travel faster than the larger ones. This separates and sorts the molecules by mass. So it's going to separate them out by mass, okay? By mass, otherwise called the flight time. So these smaller ones, they're going to fly faster. And what this does is it detects, there's a detector that records the ions. So here's my little, we're detecting and we're recording, okay? We're detecting and we're recording the ions as they arrive. So we're going to see how fast they go through and detect and record. And what happens is it generates a pattern of peaks called mass. So it's going to record it, and there's going to be some that are, some that are smaller, some that are bigger. The smaller ones are faster, the bigger ones are slower, and it's going to record all of that data, and there's going to be a pattern. So there's going to be peaks in there, and that's what's called mass spectrum, okay? This is essentially a fingerprint, okay, or a profile of proteins and other macromolecules. So you're getting, you're really learning the identity of this based on their mass and how fast they are, okay? So the proteins and other macromolecules, so you're getting a profile of the proteins and other macromolecules in the cell, particularly ribosomes. Ribosomes are very particular, okay? What's next? So let's move over here to our computer. So the computer, let me fold this in half so I can read this at the same time. So here's our computer, and the computer software then uses that profile to identify in a given microbial colony by comparing its mass spectrum to a database. I put a large database because I'm sure that thing is big. So it's comparing it to a database of results from known organisms. All right, good luck.